Good afternoon. I'm Ken Macnamara. Delighted to be back at Meeting on the Med. I was here 3 years ago with a different hat on. I was Synpromics back then, and shortly thereafter was acquired by AskBio. So I lead the team in Edinburgh and have oversight of our growing team in Europe, in Heidelberg and in Paris. In a global role, I lead our technology development programs supported. I'm going to speak to you today just to give you a brief overview of AskBio, talk about our technology toolbox and how it empowers our therapeutic portfolio. And I'd like to take a little bit of time to highlight our patient advocacy and philanthropy programs. So, at a glance, AskBio is a pioneer in the field of gene therapy. We're a company of firsts. First to clone AAV for therapeutic purposes, first to deliver intrathecally, first to treat DMD in Pompe patients. And these firsts are built on the history and heritage of technology innovation. I'd say we're committed to push the boundaries, and to do this, we keep pushing our technology toolkit. So we're working on our capsids, we're working on our expression cassettes, our promoter platforms. We're looking at immunogenicity, how to handle pre-existing neutralizing antibodies, redosing. We're looking at different delivery methods. We're enhancing gene editing tools. And of course, we're continuously improving our manufacturing process. We're leaders in the field for our clinical development programs. We have 6 programs in the clinic or going into the clinic, and a rich pipeline of earlier portfolio programs behind those. And it's underpinned by the foundation of our manufacturing process. And we have state-of-the-art facilities here in Spain and globally. We have over 30,000 square meters, to use the metric since we're in Europe, of manufacturing capacity that addresses our own internal needs, but also as a CDMO business in our Varalton subsidiary. It's a privilege to work with some of the pioneers in the gene therapy field. Sheila McHale, I can't see whether she's in the room, but she's in the building somewhere. And Jude Smolsky founded AskBio back in 2001, and Sheila's guided us over the last 20 years through the ups and downs to become one of the largest gene therapy players in the business. Jude Smolsky, of course, widely known in the field for his foundational work, seminal work in AAV vectorology, which is at the heart of the products on the market today, the AAV vectors, the self-complementary vectors, all based in Jude's early research. Kathy High leads our therapeutics portfolio. Kathy, first in the clinic with AAV in the liver, first in the clinic with AAV in skeletal muscle, and also in America in the subretinal space. Teamwork makes the dream work, as I like to tell my children, and Sheila, Jude, Kathy are joined by an experienced team. We have Tracy Dowling here today, leading our business development and general counsel. Javi Garcia is representing Varalton, and Sheila I mentioned. Okay, to the toolbox. So, AskBio continues to build on Jude's work in capsid development, harnessing that experience and the team's experience, combining it with computational biology, bioinformatics, machine learning, to increase the potency, increase the tropism and selectivity of our capsids, reduce immunogenicity, whilst also maintaining the manufacturability of the capsids. With the acquisition of Synpromics just under 3 years ago, we continue to strengthen our leadership position in expression cassette design. we have grown the Synpromics promoter platform, Prompt, increasing our control over the expression cassette, not just now on promoters, but the wider gene expression cassette, optimizing UTRs, introns, codons, controlling RNA stability, microRNA pipelines, but also now inducible platforms, so small molecules that can control gene expression, so controlling it inducibly, repressibly, and also self-regulating expression cassettes. So we're able to respond to disease triggers or cellular stress. Manufacturing is critical to our business, and Jude and Sheila invested early in developing our manufacturing platform. We have our Pro10 cell line, which scales beautifully from R&D to 2,000 litres with industry-leading yields. We have also invested heavily in our plasmid alternative, doggy bone DNA, such that we can reduce cycle times in our manufacturing, fewer contaminants from bacteria, and lower costs. Looking ahead, especially when we consider our larger indications in our therapeutic pipeline, we're developing our stable producer cell line. And a lot of this technology then comes back together with our selective promoters, where we're able to make sure that therapeutic transgene is switched off in manufacturing, where we're able to provide the control over the kinetics for our stable producer cell line. AskBio was acquired a little over a year ago by Bayer, so we're run as an independent therapy in Bayer, as an independent company. But Bayer is committed to leading the field in cell and gene therapy, and this enables us to meet our ambitions as AskBio. Although run as an independent organisation, we're able to reach through and leverage the considerable resources within Bayer, not just financial, but also the experienced clinical and translational team, the pricing, reimbursement, know-how. And in the R&D side of things, we're able to access Bayer's small molecule library, the biggest small molecule library in pharma. And we're able to combine this with our inducible promoter platform, but also within our AAV formulations and our gene editing tools. The platform's strength has resulted in numerous acquisitions and collaborations. Recently announced our joint collaboration with Bayer and Mammoth Biosciences in the gene editing space. We acquired Brain Vectis and BNB to grow our leadership in CNS gene therapy. And in redosing, we're working with Selecta and Hansa to address some of the immunogenicity challenges we face. This toolbox and integrated AAV platform enables us to work all the way from monogenetic to pathway diseases. So we take our learnings from the orphan disease programs and are managing to bridge the gap to the larger indications. Our portfolio, we have reflects that. We have Pompe disease, congestive heart failure, and Parkinson's disease in the clinic, and expect to be going into the clinic with 3 more programs this year with limb-girdle, Huntington's disease, and MSA. Beyond that, we have a number of early-stage programs in the heart, in muscle, in liver, and in blood diseases. So we continue to innovate. In our manufacturing process, through process development, cell line engineering, we continue to improve our yields. With our promoter and regulation platforms, we continue to improve safety for the patient and also enable new market opportunities with that greater level of control. With doggy bone DNA, we're shortening our development cycles, getting our therapies to patients faster and safer. And we're tackling the issues on immunogenicity, redosing. the fact that we have all of these technologies under our own control gives us resilience. It gives us a plug-and-play platform, and we're able to accelerate as many programs as we are. Okay, I realize I'm between this room and lunch, but I did want to mention our Ask First patient engagement program. The collaboration program is a group formed of advocacy groups, patients, families, and is at the core of our decision-making process in our evaluation and development programs. And I would also like to mention the CCF, the Columbus Children's Foundation, founded by Jude and Sheila here in Spain back in 2017. As a business, and as the field grows, we wanted to make sure no patients were left behind. So the CCF Foundation is continuing work on ultra-rare diseases, particularly in children. And we've now dosed and treated almost 30 patients, 30 children, for AADC deficiency with remarkable transformative results. So to sum up, our experience and know-how in our science and technology, our experience and know-how in our clinical programs and manufacturing puts us in a position to accelerate a very broad portfolio of programs and keep pioneering the space that AskBio is known for. Thank you.